General properties of viruses. Introduction. Viruses are smallest known infective agents. They are the simplest form of life seen, and they are having a single cell-like structure, but they are not strictly unicellular organisms, because in even the simplest of unicellular organisms has its own machinery, its own setup for carrying out all the metabolic processes. But viruses do not have a cellular organization. They are incomplete structures, and they depend on the host cells for various resources. Main properties of viruses include that they do not have any cellular organization. They have either DNA or RNA. In general, most organisms they have DNA and RNA both, but viruses have only one of them. They are obligate intercellular parasites. They lack enzymes for synthesis and replication. They multiply by a complex process, unlike bacteria, which multiply by binary fission. Viruses are unaffected by antibacterials. Antibiotics do not have any effect on viruses. The morphology of viruses. Viruses measure in nanometers. They are much smaller compared to even bacteria. And example of smallest virus include parvovirus, which is as small as about 20 nanometers. And example of largest viruses include poxvirus, which is as much as 300 nanometers. The extracellular infectious virus particle is known as virion. Viruses were given various names by scientists, like they were called filtrable viruses because they could pass through bacterial filters. They were called ultramicroscopic as they were not visible in the light microscope. They were also called elementary bodies, large viruses which were stained like pox virus. They could be seen in the light microscope, and they were given the name elementary bodies. For measuring the size of viruses, scientists have used various methods like colloidal membrane filters, electron microscopy, sedimentation in ultracentrifuge, and comparative measurements. Here there is a figure in which bacterium Staphylococcus aureus is shown, a 1 micrometer big Staphylococcus aureus, and compared to this 1 micrometer Staph aureus, chlamydia are very small, as you can see in the figure. Although chlamydia are bacteria, but they are very small structures. Then the largest viruses like pox virus, compared to Staphylococcus aureus, they would look so small. And there are viruses much smaller than that as well, like herpes virus, influenza virus, and poliovirus. So you can see here the bacteria which is in micrometers would look so big compared to the viruses which are in nanometers. This is a table which shows different sizes and different kinds of cells falling in different size groups. Starting from 1 centimeter to 1 millimeter, 10 to 10 raised to minus 4, 10 raised to minus 5, then 1 micrometer, then up to 1 nanometer, and up to 1 angstrom. So in this size range, you can see that animal cells fall in the category of about 10 raised to minus 5 meters. Bacteria fall in the category of micrometers, about 10 raised to minus 6 meters. Viruses are smaller than bacteria, they fall in nanometer category. Proteins are smaller than viruses, and atoms are the smallest of all, measuring in amount angstroms. In the electron microscope, some viruses will appear in icosahedral shape. You can see here an example, rotavirus and adenovirus. Various shapes are there in viruses. Many viruses occur in spherical shape. Some of them are irregular, some of them can be pleomorphic. Some peculiar shapes found in viruses include bullet-shaped virus, example rabies virus, brick-shaped virus, pox virus, rod-shaped virus, tobacco mosaic virus. Here we will see some figures which show some viruses in the electron microscope. Spherical virus like adenovirus, rod-shaped virus, example tobacco mosaic virus, brick-shaped virus, example pox virus, tadpole-shaped virus, example bacteriophage, bullet-shaped virus, example rabies virus, and filament shaped virus, example Ebola virus. So these are various shapes found in viruses: brick shaped, bullet shaped, filament shaped, rod shaped, etc. Now, what is the structure and chemical composition of viruses? To understand this component, we understand it in 4 categories: viral capsid, viral symmetry, viral envelope, and viral nucleic acids. In all the viruses, there is a nucleic acid which is at the innermost portion. The nucleic acid is surrounded by a protein coat which is known as capsid, and in some viruses there can be an additional covering outside the capsid which is known as envelope. Envelope is an optional property which is there in some viruses and not there in some viruses. So some viruses are enveloped and some are non-enveloped, but 2 portions, that is the viral core, that is nucleic acid, and the capsid, these are there in all the viruses. So this together, the nucleic acid and the capsid together are known as nucleocapsid. Nucleic acid plus capsid is the nucleocapsid. The capsid is made up of capsomeres, which are made up of polypeptide molecules. So the nucleic acid is surrounded by a protein covering known as capsid, which is made up of capsomeres. In this figure, we can see some viruses. First one is tobacco mosaic virus. It has a helical structure, helical rod-like shape. The RNA inside is coiled in a helical manner surrounded by a capsomere. Then adenovirus is shown in the icosahedral shape with the DNA inside surrounded by a capsomere. Influenza virus has multiple layers. The innermost is the RNA. The RNA is surrounded by a capsid, and the capsid is further surrounded by a membranous envelope. The envelope has glycoprotein spikes on it. So influenza virus is an example of envelope virus. Then you can see bacteriophage, which is a tadpole in shape. It has a head, it has a tail, and it has the DNA inside the head. Now, what are the functions of capsid? Capsid is the protein coat which surrounds the nucleic acid. Together, the nucleic acid and the capsid form the nucleocapsid. The capsid is covering the nucleic acid, so it protects the nucleic acid. It acts as a binding site. It facilitates assembly and packaging of viral genetic information. It acts as a vehicle of transmission. It has antigenic properties. It will activate the host immune defenses, and the viral capsid also provides a structural symmetry to the virus. Different kinds of viral symmetries are there. 2 common symmetries observed are icosahedral symmetry and helical symmetry. Besides these 2 common symmetries, there are many other varieties of symmetries which fall in the category of complex symmetry. So there are 3 kinds of symmetries: icosahedral, helical, and complex. First is the icosahedral symmetry. In the icosahedral symmetry, there are 12 vertices and 20 facets. This kind of a structure with 12 vertices and 20 facets is known as icosahedron, and some viruses occur in the icosahedral shape. Example, adenovirus, as you can see in the microscope. Helical symmetry. In helical symmetry, the genetic material is coiled in a helical manner inside. It is surrounded by a capsid, which is also surrounded in a helical manner. This kind of symmetry is called helical symmetry. Then third is complex symmetry. All other symmetries which are not icosahedral and helical, they fall in the category of complex symmetry. Example, T4 bacteriophage. The viral envelope is made up of glycoprotein. It has lipid from the host cell and protein from the virus. Now, viral envelope has lipid, and so they are susceptible to lipid solvents. All those chemicals which dissolve lipid, they will be effective against envelope viruses, but they will not be effective against naked viruses as they do not have a lipid envelope. Envelopes confer chemical, antigenic, and biological properties to the viruses. And in many viruses, the envelope is having some protein spikes. These protein spikes are projecting outside the envelope, and Example of such protein spikes include hemagglutinin and neuraminidase. And functions of these protein spikes include mediating attachment to the receptors, enzymatic activity, and they act as major antigens. The protein spikes are also known as peplomers. Here we can see a figure which shows a naked virus and an enveloped virus. The naked virus has only two components, the nucleic acid inside surrounded by the capsid outside, while the enveloped virus has three portions, the nucleic acid in the innermost surrounded by the capsid, which is further surrounded by the envelope, and the envelope has various protein spikes like hemagglutinin spikes and neuraminidase spikes are examples of these protein spikes. The spike is also known as peplomer. Here 6 DNA viruses have been enumerated in this table. 3 of them are envelope and 3 of them are non-envelope. The envelope DNA viruses include Pox viridae, Herpes viridae and Hyphaidna-viridae. The envelope DNA viruses include Pox viridae, Herpes viridae and Hyphaidna-viridae, while the non-envelope DNA viruses include Adenoviridae, Papoviridae and Parvoviridae. So 6 DNA viruses are there, 6 families of DNA viruses are there, out of which 3 are enveloped and 3 are non-enveloped. There are various RNA viruses, and in this table some examples of enveloped and non-enveloped both kinds of RNA viruses have been shown. Examples of enveloped RNA viruses include Orthomyxoviridae, Paramyxoviridae, Erinaviridae, Bunyaviridae and Rhabdoviridae. Orthomyxo, Paramyxo, Erina, Bunya and Rhabdo are examples of enveloped RNA viruses, while non-enveloped RNA viruses include Caliciviridae, Picornaviridae and Rioviridae. Calici, Picorna and Rio are examples of non-enveloped RNA viruses. The viral nucleic acid is either DNA or RNA. It can be single stranded or double stranded. It can be circular or linear. It can be segmented or non-segmented. Based on this, the viral nucleic acid can be categorized as DNA or RNA. The DNA can be categorized as double stranded or single stranded. In both double stranded and single stranded, linear or circular, and it could be single chain or multiple chain DNA. Then the RNA can be divided into double stranded and single stranded. The double stranded RNA has single or multiple chains, and the single stranded RNA has positive sense or negative sense, 2 types, and in both of them there can be single or multiple chains. With this we finish General Properties of Viruses Part 1.